Hello, and welcome to this webinar titled Developability Assessment of Therapeutic Proteins, a Toolbox for Tackling Increasing Complexity, hosted by Biopharma Asia magazine and presented by Thorsten Lorenz, Team Head of Developability Assessment Biologics at Novartis, and Beata Kern, Product Manager, Nanotemper Technologies. My name is Stephen Edwards, and I will be your moderator. Before we begin, I would just like to remind our viewers that there will be a live Q&A session at the end of the presentation. Audience members can send their questions in at any time during the presentation via the Questions tab located directly below your webinar screen, and we will go through these at the end of the webinar. Now, please allow me to introduce our first presenter, Thorsten Lorenz. Thorsten Lorenz studied biochemistry at the University of Bochum in Germany. He performed his PhD work at the Max Planck Institute for Medical Research in Heidelberg, and afterwards joined the Novartis Institute for Medical Research as a postdoctoral fellow. In 2011, he joined the Integrated Biologics Profiling, or IBP unit, at Novartis, setting up and leading a laboratory for biophysical characterization and pre-formulation assessment, and got appointed group head in January 2016. In his current role as head of developability assessment, he is responsible for the assessment of developability characteristics, including molecular properties, downstream process and pre-formulation aspects, immunogenicity risk, as well as in vivo fitness for all biologics of the Novartis pipeline at the transition from research to development. I will now be handing over to our first presenter. Welcome, Thorsten.
Thank you very much, Stephen, for this very kind introduction, and welcome everybody to our first part of today's webinar. I will be talking about the developability assessment of therapeutic proteins and provide you a few examples of our toolbox concept to address a variety of different molecules that we do have in our pipeline. But before I get into the details, I would like to remind everybody here that these slides are intended for educational purposes only and for the personal use of the audience. These slides are not intended for wider distribution outside the intended purpose without my approval. Furthermore, the content of the slide deck is accurate to the best of my knowledge at the time of production. So what I would like to present you in the next 25 minutes is also relating to a publication that we recently published in the Biopharma Asia journal, the May-June issue. So you also will find several of the figures in this publication, but I also would like to go a little bit more beyond and show you several examples how we, besides having an overall developability assessment strategy, also applies it with specific examples. And here we will give you examples from the purification screening and how we also apply automation concept there to really deal with the high number of samples. I also will go a little bit more in detail on the analytical methods toolbox, in particular also the need for the optimization of methods. And then also we'll take an example on how we apply high-throughput stability testing. And last but not least, there will also be a few slides on our pre-formulation concept and also an example on how we apply high-throughput viscosity screening. So with this, I would like to give a brief introduction on what we are doing. So Integrated Biologics Profiling at Novartis combines several technical functions, starting basically from a cell line development organization up to the formulation assessment early on. The Developability Assessment Group itself is responsible for several areas, contributing to the early assessment of all kinds of biologics. However, our primary focus, just based on the pipeline, is currently protein formats, but here we have a broad spread of different molecules, basically standard antibodies, through bispecifics, conjugates, Fc fusion, enzymes, and other kind of molecule formats. Just very briefly, the group. So as I mentioned, we are involved in early screening of candidates. A lot of automation is applied there to really deal with a high number of samples. But then we are also getting into the purification of the material, which can go up to gram scale amounts, and I will come to this in a minute. But besides providing the material, a very important part of our purification scheme is also the process assessment. From a characterization point of view, we have a lot of physicochemical characterization, and I will show you the toolbox later. But importantly to mention, this is also often forgotten, that the data that we collect throughout the in-process control analytics are an essential part of our overall integrated assessment. Mass spectrometry analytics becomes, of course, more and more important because the questions are getting more complex, and to identify the respective potential liabilities require an in-depth characterization via mass spectrometry. Additionally, we also include immunogenicity risk assessment and also an in vivo stability assessment to really identify potential risks that may come up when the molecules are later on injected into the human body. And last but not least, one of our major work pages is also in the formulation and solubility screening. All this is combined in an overall candidate selection process, material production, and an overall risk and effort assessment that we provide with a report to our research project teams as well as to our development organization. So let's start with an overview on what the phases typically look like for the developability assessment IDP. And even though I here start with the standard antibodies, the phases that we defined in screening, profiling, and lead development, which happen prior to the final lead selection, are more or less looking the same for any kind of protein molecule that we are dealing with. The advantage for the antibodies is a long history of experience with those molecules, basically allowing us to funneling down the candidate numbers from typically a high set of candidates, 50 to 100 candidates when we're starting with the screening, into the final lead candidate. And the importance here is due to the strong knowledge that we do have around our antibodies, that we are not only assessing the molecule if they are suitable, but we are really trying to drive them into a platform fit, meaning that the later processes from upstream through downstream processing, analytical characterization, and also the formulation do fit into our platform to really be efficient and fast throughout the development process. On the right-hand side of this slide, you see this funnel that I was just mentioning. So starting off typically with 50 to 100 candidates, we have a very streamlined high-throughput expression technology using transient tech in the early stages and only applying a very few analytical assays which are predictive for the liabilities that may come up. And ideally at this point in time, we already deselect the ones that do have severe challenges with respect to aggregation or maybe increased hydrophobicity. During the profiling, we go more into depth, and we are also increasing the scale. So we are going into the stable choke production, the final cell line, and we are producing around 200 milligrams, which serves our needs for characterization, but can also be provided to the research project teams to do initial biological assays. Based on those data, we finally select the 2 lead candidates, which we then bring into larger bioreactor production to provide usually around 50 to 80 grams of material, applying more intensive analytical characterization, up-concentrate the material typically higher than 100 or 150 milligrams per milliliter, and then also apply the pre-formulation assessment package and solubility, which is only possible at this point in time due to the high material demands. On the contrary, the developability assessment for therapeutics is more custom-made and more dedicated to the individual molecule that we are dealing with. The phases are basically the same, but it's here the situation that we typically have only a few candidates entering already the screening phase. At the same time, it's not given that we directly go into our standard CHO cell line, but in most of the cases, we do a broad multiplexing of different cell line or strain combinations, including vector multiplexing. Also, the purification process is not standardized, so there's a lot of work that goes into to have a meaningful purification process to deliver high-quality material at the end of our process. Along the same line, also the formulation development activities as well as the characterization are very candidate-specific and need to be tuned every time. And likewise, also here we are not aiming for a platform because there is no platform for the diversity of the different molecules, but we are rather here providing a design space and a risk-effort assessment to enable performance. So it's not a funneling down to the final lead candidate from a high set of molecules, but rather really to make sure that the few candidates that we do have are well characterized, understood, and also enabled from a technical point of view. And that we are also early on engaging with the development functions to really bring together all the data and to have a data-driven decision on the development path. On this slide here, I just want to highlight, and this is also in the publication, how we separate out the different phases. So, as I said, in the screening, we are very much focused on finding the right early conditions. And often for therapeutic proteins, we do see that the titer is the major challenge for the further steps. So therefore, the multiplexing of cell lines are typically aiming to first boost the titer to a meaningful level. However, if we also do see throughout this process that degradation might arise, proteolytic degradation, then we also can adjust the cell lines and also trigger potential re-engineering to optimize the molecules with regard to those effects to have a meaningful purity before we start into the purification process. And this we can also follow into several cycles. If also then aggregation might be a challenge, we also might specifically address this. And also the analytical strategy is designed in this direction so that we really focus individually and not blow the resource needs to do everything early on before we have a meaningful first quality of the material. During the profiling phase, we are going more into depth. So there we will then have a set of molecules and also a suitable cell line or an expression host selected. And there we then tune the package according to the project needs. So here I just listed, I should also click, apologies for this. Here I also listed different kinds of proteins where you would see that in some cases you might have a higher need to invest more work packages into optimizing the cell line part, whereas in other project types you might really have a strong need for investing more into the analytical work packages, as you can see for TP2. And TP2 could be, for example, an antibody drug conjugate, where the purification process up to the conjugation is typically more like a standard antibody process. However, when it then comes to the further characterization in the presence of the linker payload, you do need to assess more characteristics, and therefore the analytical work packages are increased. Last but not least, when we then take the final two lead candidates throughout the lead development process, we're really focusing on improving the manufacturing process to a certain extent to be able to deliver high quality material in gram scale, as mentioned before, for example, for dose range finding studies, but also to have a very solid purity of the material when we are entering the pre-formulation assessment studies. And at the end of this, we do have a thorough report which comprises all the data collected and assessment done throughout the screening up to the lead development. This slide here gives a very quick overview of the expression toolbox that we are applying. It's important here to mention that we are not only looking into mammalian host cells, but that we also have the possibilities to look into other possibilities like microbials in general. And that also, if we do not have appropriate host identified from the get-go, that we also can invest into custom cell lines and improve further from there. Of course, this has implication on timelines and needs to be very specifically defined with the project teams as we go through the process. Also, in combination with the different vectors and different candidates in the set, you see that the number of samples early on, despite the relatively low number of molecules, can increase quite significantly. So on this slide here, I would like to briefly go through the purification screening. And this is important because for standard antibodies, we typically have a very straightforward purification process, as you can see on the left-hand side here. So an affinity capture of the clarified harvest, a virus inactivation step before we then go into the preparative polish, which in really the utmost cases can be done by a standard cation exchange chromatography step. And then we go into buffer exchange and up concentration of the material, which is used for the formulation studies, and a further formulation for respective in vivo studies. However, for most of the therapeutics, the situation is not as easy, but assuming that we have a molecule with an affinity tag, the capture usually is a protein A step, and we can easily go through up to the polish step. However, before we go in there, we already have an idea of how the molecule might behave based on its PI, based on prior experience in the screening phase. And based on this, we then design the polish screening, which is shown here on the right-hand side. So depending on the PI, as said before, and the observations, we already might discard certain conditions, but all others are then broadly screened to find the best conditions for the preparative polish screen. Once this best condition is identified, the preparative process is continued and the material is formulated as mentioned before. What I also would like to highlight here is the importance that the number of samples also in this stage can very quickly expand. Assuming that we have 3 different salt conditions at 3 different pHs and we would like to test 4 different resin types, we already end up at 36 test conditions. And this, of course, already here may impact significantly the material needs, also the hands-on time for preparation of equipment and buffers, the runtime for the samples, and what also our analytical colleagues are always saying, of course, the number of the analytical samples for the in-process control analytics. And therefore, to really streamline this, we recently introduced a robotic system to perform these kinds of screens in a high throughput manner, and which is also predictive for the preparative scale later on. So we perform our DSP screenings on a Tecan workstation, which carries out all the sampling, handling, including the centrifugation steps, pH measurements, UV and conductivity measurements, and of course the preparation of the analytical sample plates. So the first step, as you can see here, the intended test conditions are entered into a wizard, which is then converted by the system into a working instruction for the robotic system. The different proteins are loaded onto different equilibrated resins, and the individual fractions are collected throughout the loading, elution, and the washing steps. The whole plate is subsequently measured for the actual pH, the conductivity, and the UV absorption for each and every individual well, which, as you can see here, basically results in a profile which is very well comparable to an online measurement. The very neat thing is that we here then can select specific fractions that we want to subject to further process control analytics. And this can be done from the recorded data sheet and then automatically picked by the robotic system, transferred into the analytical master plate, and then submitted to the respective analytics that we would like to apply on these samples. So all collected data are in the end used to select the best conditions for the preparative run, which is then performed typically on an Agda system to yield the respective material needs. So what we have recognized is that this set has significantly improved our efficiency to really screen a broad set of conditions to find optimal candidate-specific purification conditions. As I mentioned before, analytics plays a role in all these steps that we are looking on early on, and therefore also from an analytical perspective, we need to distinguish how we run the support. So typically we separate those methods into in-process control analytics methods, and here titer and concentration are very broadly used. But then also purity, clipping, and fragmentation methods are applied. When it comes to then bispecific molecules, for example, also we include mispairing tests. Aggregation is a very typical one, and then also in specific formats like conjugates, we are also looking into the drug-antibody ratio, for example. And then if you break this further down into the individual techniques, these are ALC, reverse phase methods, typically size extrusion chromatography and UV spectroscopy. And similarly, also we handle this for our work packages with regard to the biophysical profiling. So here, typically, we are looking at the hydrophobicity, at the conformational stability. Viscosity is a very important aspect, especially when we are going into high concentration liquid formulations. The stability is looked on early on, self-interaction, and the methods to this are given here on the right-hand side. And last but not least, the quality check is also then going more into the detail of the molecule, and which is also process-dependently influenced, and therefore purity is also checked at the end of the process. But also then post-translational modifications are very closely monitored with regard to deamidation, isomerization, oxidation, and also serialization, and also other aspects might show up depending on the molecule's properties. What I also would like to mention here is that we have a standard set of assays, but for therapeutic proteins, we, in most of the cases, early on enter also an intensive method development to really have the appropriate quality of the methods to characterize these molecules. In this example, I would like to give you on one of our recent projects, which is a conjugated protein, which already entered with quite some challenges for the process. It had a relatively low PI, which brought a challenge on the DSP process. It was highly hydrophobic, and we had 2 conjugation sites, and both could be filled or not. And this also influenced the way we could set up the in-process control analytics. And here's the example of the size exclusion chromatography, and the chromatogram on the upper left-hand side. I highlight this in red because you cannot really call this a chromatogram. It has a very poor recovery, and also the resolution was basically not existent. So there we needed to invest quite a bit into the optimization of the method, and this is you see in the middle of the scheme. So this is an iterative process that was applied. We first looked at different mobile phases and also screened different kinds of resins, which already led to a very meaningful recovery, as you can see in the first figure in the yellow box. But then we also worked on the pore size screening and the particle size screening. And then also, since we knew that we were dealing here with a hydrophobic molecule, we also tested specific resins with the respective particle and pore size, which was specifically designed for hydrophobic molecules. And this ended up already in a very good recovery and improved resolution. And 2 more rounds in optimizing the mobile phase and also adding organic modifiers really ended up in a high resolution method with full recovery of the molecule, and it really could enable the purification process for this kind of molecule. Another example that I wanted to mention here, and that you also will hear about in the second part a little bit more of this webinar, is also how do we ensure that we can screen broader conditions early on to find the best conditions, and not putting our bets for a specific molecule on an inappropriate buffer condition. And one possibility here is to use the conformational stability of a molecule to test out increase or decrease in the melting temperature of proteins, which we apply at different stages of our process. So we can usually apply this already in the early screening phase, but we are also using this when we are optimizing the conditions for the formulation screening later on. In this example here, protein X is one example where we optimize the conditions for our pre-formulation assessment. So we wanted to see how the molecule behaves in the different pH ranges in the presence of different salts and excipients. And we also wanted to save as much as material as possible. And here you very nicely see the transitions of one molecule in different buffer conditions, which ranged pretty broadly and also indicated that certain conditions even induced aggregation upon unfolding. The replot of these data is shown in this graph here. In the upper panel, you do see the TM, so basically the maximum of the first transition. And also we looked at the unfolding transition, because depending on the sharpness or the breadth of the peak, this can have also additional information when it comes to stressing the material later on. And here to highlight is that we saw a significant stabilization of protein X in the presence of excipient or ionic strength. So both had a very positive influence, and that also we could identify the unfavorable pH region, which was exactly at the edges of our screen here. So this we basically used to screen more closely around the middle pH ranges in the presence of ionic strength, and could also exclude conditions from higher pH, lower pH, as well as in the absence of salt. I mentioned before, these kind of techniques, as well as the whole process, also in the end funnels into a pre-formulation assessment, where we also generate a large set of information in terms of stability and solubility of the molecules. But also here, we try to separate the kind of complexity of the molecule to ensure that we are investing the right amount of resources at the right time. And as I said before, the antibodies, we are really typically aiming for a platform formulation, and therefore we have a very defined set of conditions that we are looking at. which we call platform formulations, with certain buffer conditions around these specific platforms. We use fully formulated samples, and we also include a 3-month stability for confirmation of our findings upon accelerated stability. And basically, once we can confirm in this set that the platform formulation is fitting, this formulation can already be locked for the clinical service form. For medium complexity molecules, we basically treat it very similar. The thing here is that we do expand the set of conditions slightly at the edges of the pHs. We also might include additional salt conditions if required. But basically also we go in with fully formulated sample. We also have a 3-month stability confirmation. And we are also in the position to lock the clinical service form if we identify a fully formulated sample without any liabilities. The situation, again, for the higher complexity molecule is different. We have a fully flexible design because, as I mentioned before, there is no platform fit for the diversity of molecules that we are dealing with. And we are doing a lot of pre-screening with regard to TM and solubility to get the best design space for our formulation study. To also reduce the number of conditions and still get a very strong readout, we are using a design of experiment approach to cover a broad range of conditions. And we are typically not using any conditions such as sugars or surfactants in our formulations to really identify first the liabilities and only in the next step of the process then optimize further with regard to the respective excipients. So this is a staggered approach where we have our first bit in IBP with the liability screening and candidate selection, and then the pharmaceutical development group takes over to really go into the depths of the formulation development. So here we do not lock any formulation for the clinical service form, but we have seen that this will not be possible without further investment in the pharmaceutical development. And this is also a figure that you find in the publication, and it's really meant to very high level show the information that goes in and also how the conditions are set up to come up with a final interpretation of the risk and the effort that goes along with the molecule. So since when we start with a pre-formulation assessment, there's a huge bunch of information that can already feed it into the design of the study. So we have a lot of project-specific information like the indication or the route of administration that we are going to go. So are we aiming for a high concentration or rather low concentration? We have the process-related information from the upstream and the downstream process, where we also may already know that certain buffer conditions might lead to precipitation. We have all the biophysical properties and also the solubility screens that run before to avoid conditions that might not be feasible to test further. So with this, we set up this stress conditions with regard to pH range, protein concentration range, ionic strengths, different temperature and mechanical stress conditions. And then again, depending on the needs of the project, we also may include stresses like photostress already at this stage of the project. Then the analytical readout basically take all the methods of the toolbox that I was describing a few slides before. And then we also provide a risk effort assessment from a formulation point of view, and also give a recommendation for the molecule based on the formulation assessment. However, this also feeds into the overall report, where all the information comes together with a final conclusion on the recommendation. This is my last slide before I come to the summary, and I see that I'm already a little bit over time. But this I really wanted to show because we have made a huge advancement to also be able to really screen a lot of the conditions with regard to viscosity. And this is just because of the fact that more and more projects do require high concentration liquid formulations. And the molecules often show the risk to have increased viscosity, which we definitely want to avoid for the molecules when taken forward, either by taking a more preferential molecule or by having the possibility to control the viscosity with the addition of the respective excipients. And this is exactly what we are trying to screen also during the developability assessment. So this is just one protein Y here, but typically we have more proteins that we apply to this, and then we are testing different buffer conditions in the presence or absence of ionic strength, and also in the presence or absence of viscosity reducing agent. And this is just an example that nicely shows how important it is to also look at the different pH conditions and not only applying a concentration range for the protein. So here we tested 35 conditions with regard to protein concentration, pH, and excipients, which could be tested within 24 hours with minimal hands-on time. We nicely see that the low pH range positively affects the viscosity, so conditions A and B were basically the ones which did not show any challenge with regard to viscosity. The red line that you see is what we typically set as a threshold for the viscosity, so all conditions stayed below the thresholds. And that when we increased the pH or the viscosity, especially in the conditions for only buffer, increased significantly. And if we went even further, besides the viscosity challenge, we also saw that the molecule started to precipitate, and therefore these conditions could be later on excluded, and we could again focus more on the more preferential conditions. So in summary, I hope I could show a little bit that an early and systematic developability assessment during lead discovery and the optimization phase is able to select the best candidates with a desired developability profile and to really identify the most suitable lead molecule, and also potential risks that we might need to address in the further process, so a data-driven decision. We usually use the format experience and the respective project needs to really design our developability assessment in the individual phases that I described. And that also it is important to have an adequate early method in place and the process development, which is essential to have a good assessment of the individual molecules. A broad set of conditions becomes more and more important. This also leads to the last bullet point. Also, the way we are applying our assessment goes more and more in the direction of high-throughput applications, automated applications, and also the use of design of experiment processes to really be able to evaluate the large number of sample sets. So what I was telling today, a cross-section through the different aspects, is of course only possible with a contribution of a very large number of people, dedicated and excellent scientists. So I would really like to thank the people highlighted here on this slide and my colleagues in various research and development functions. And in the end, I also would like to thank you for your attention. With this, I hand over. Thank you very much.
Thank you, Thorsten. Once again, I'd like to remind our viewers that there will be a live question and answer session at the end of the presentation. Please feel free to send in your questions at any time during the presentation question tab located directly below your webinar screen, and we'll go through these at the end of the webinar. Now, please allow me to introduce our second speaker, Beate Kern. Dr. Beate Kern studied molecular biotechnology at the Technical University of Munich and the Tokyo Institute of Technology. She went on to her PhD in medical microbiology at the Ludwig Maximilian University of Munich, where she studied Helicobacter pylori and its effects on human T cells on a molecular level. Beate joined Nanotemper in 2015 as an application specialist in the customer support team. Currently, she is a product manager for the Prometheus and Tycho product lines. I will now be handing over to our second presenter. Welcome, Beate.
Thank you, Stephen, for the introduction, and thank you also to Thorsten for this great overview of how you approach developability assessment at Novartis. So we've already seen how Novartis uses the Prometheus in their workflow, and now it's my pleasure to give a broader intro to protein stability and to the Prometheus. All right. I'm sure that most of you are familiar with the concept of protein stability, but it isn't always clear to people what all you can use protein stability information for. So I just want to use thermal stability as an example to highlight some of the possibilities that you have, how you can use this. So for example, you can use the thermal stability of a protein as an indicator of the overall stability. This is what Thorsten showed in his example. You can use the thermal stability of a protein to indicate which one will be the optimal formulation, the optimal buffer conditions, storage conditions, excipients, pH, and so on. Of course, you can use thermal stability to characterize your protein structure. So this is a different way of looking at it, to look at its structural integrity. You can look at different domains of the protein if you have a sensitive enough method like the Prometheus is. You can use it for biosimilar profiling for the same reason. If you have a good resolution method, you can use thermal stability to look at the similarity of different proteins. And of course, you can also use it for finding protein variants that are suited for very specific applications. For example, if you're engineering enzymes and you need an enzyme that can tolerate very high temperatures or organic solvents or something like that, then thermal stability can also be very helpful. Now, the Prometheus system that I'm introducing today does not only look at thermal stability, that is its main application, but it can also help you look at thermal unfolding and refolding kinetics. It can help you look at chemical stability, and it can also help you look at aggregation of proteins. And all of these are, of course, different aspects of protein stability that can be very helpful if you really understand them. Due to the unique way of how Prometheus does its measurements, that I will of course talk about in detail later, it can be used anywhere in the biologics workflow, and it can really help at any of those steps. So Thorsten showed an example of where Prometheus was used early on in early screening on rather smaller scale, where you have low concentrations of protein, low sample volumes, and so on. So this is where it's useful, but you can also use it later, for example, for formulation screen, because it can also deal very well with high concentrations and high viscosity samples. Now to talk about some concrete example applications, I want to first give a very quick overview of how the measurement works and what the data usually looks like. So this is what you can see here. We use intrinsic protein fluorescence in the Prometheus to monitor the protein unfolding process. The reason why this is possible is that the tryptophan residues that contribute most significantly to the fluorescent signal, they are very sensitive to their environment. So in the folded state of the protein, the tryptophan residues are typically buried inside the core of the protein, but then upon unfolding, as you can see here on the top, during unfolding, the tryptophan residues become exposed to the buffer, and therefore their fluorescence changes dramatically. And this is what we can see. And the kind of signal that we typically get out is shown at the bottom. This is a generic example, of course, and it can look very different too. But a lot of the time, and also in Thorsten's example, it looked like this. You start with a low fluorescence signal for the folded state of the protein, and you finish with a high fluorescence signal of the unfolded protein. Depending on the type of method you want to use, so how you unfold your protein really is, of course, your choice. So if you're looking at thermal stability, you would use a temperature ramp to unfold the protein. If you're looking at chemical stability, you would use a denaturant at varying concentrations to unfold your protein. Then, of course, the halfway point of this process is what typically interests people. In thermal unfolding, this is the TM. In chemical unfolding, this is CM or C50. This is the point where half of your protein is unfolded. There are more parameters that you can get. For example, Thorsten also mentioned the onset of unfolding that can be very helpful, but the TM is the most typical one that people are looking for. Now, in a concrete example, I want to show what that can look like in a real example, where we are looking at an antibody, and you can already see that the unfolding profile of this antibody looks a little bit more complex than what I showed in the previous slide. So instead of just having the one transition, we can see two here, two steps in the profile. This is, of course, due to the fact that antibodies have several domains that are linked rather loosely. So the unfolding of one domain doesn't necessarily influence the unfolding of the other domains, and that's why we get separate unfolding events that we can see in Prometheus due to the great resolution that we have. For this example, this is important because we are looking at an antibody before and after conjugation to a drug. This is, of course, something that is done routinely, and you might want to understand how the conjugation of your drug to the antibody affects the antibody stability. And you can see very clearly here that the second unfolding event, the one at higher temperatures, around 86 degrees or something, is the same for the naked antibody as well as for the ADC. It happens at the same temperature. But if you look at the first unfolding event happening here around 70 degrees, you can see that there is a difference, quite a clear difference, between the naked antibody and the ADC. It's about a 2-degree difference or something. And this, of course, can already be very significant. Now you know that conjugation of the drug to your antibody destabilizes this domain, but not the other one. And then it's up to you to decide, of course, if this is a problem, or if this is okay, or if this is better than you expected, of course, depending on your situation. We can also look at this type of data in a different way. I just want to highlight this because it makes it a little bit easier to visually interpret. So instead of looking at the fluorescence data directly, we can look at the first derivative of this, meaning that instead of these steps and transitions, we get peaks. And these are a lot easier to identify visually. So you can very easily see that, again, the higher temperature unfolding event happens at the same time for the naked antibody and the ADC because the peaks overlap very nicely. Whereas for the lower temperature unfolding event, you can see that the peaks are shifted. And that clearly indicates that this domain is being destabilized by the conjugation. In another example here, I want to talk about biosimilar profiling. Prometheus has gives you the advantage of being able to screen many candidates early on, making it possible for you to weed out those that aren't promising. Whereas, of course, during the development of the biosimilar, you have to use many technologies, many methods to really prove the similarity of your candidate to the originator and to really show that it's functional and so on. But all of these methods are very elaborate, time-consuming, costly, and so on. So you don't want to waste time and money and effort on those samples that aren't worth it. And Prometheus can help you screen for those things early on. In this specific example, we worked with UGA Biopharma to look at 16 biosimilar candidates and compare them to a reference. And you can already see here very clearly that they look quite different. These are all the same, of course, as it is with biosimilars, all the same sequence, but expressed in different systems, and they look very different in their thermal unfolding profiles. We found a best match to look very similar to the originator. You can see that here in the top center panel. And of course, we also found a worst match. You can see the difference very clearly in the bottom center panel here. And as I said, this can just help you to get rid of that, of those samples that look like this worst match, for example, get rid of them early and not carry them through to later stages. The next example I have is a little different. So here we're not looking at simple thermal stability, but we're looking at a different way of looking at this by understanding the unfolding reversibility of a molecule. You can really get more deep into how your protein stability works. So for this example, we looked at 2 antibodies in what we call an incremental temperature cycling experiment. In the image on the left, you can see a dotted line in the background. This is the temperature profile that was run. So you can see that we start at a low temperature. Then for the first cycle, we increase the temperature, but then we come back down to the base temperature. Then for the next cycle, we increase a little more and then again come back down to base. And then we keep doing this until we reach very high temperatures, but always coming back down to the base temperature. The blue line in this image is the unfolding profile of this antibody. And you can see that for quite a while, nothing really happens. We have a flat line for the first few cycles, sorry. This means that the antibody is not yet unfolding. That's fine. And then at some point, you can see a little bump, and the bump gets bigger for the next few cycles. But the fluorescence always comes back down to base, or pretty much comes back down to base, meaning that the unfolding that is already happening here is reversible. Now, of course, at some point, this is not true anymore. At some point, unfolding becomes irreversible. The signal does not come back down to base, and we know now we're really at a place where we can't go back. In comparison, for a second antibody, the picture looks very different. We applied the same cycles of temperature increases here, but you can see that from the very beginning there is unfolding, and also from the very beginning unfolding is irreversible because the signal never comes back down to the original initial signal that we started out with. So for this antibody, all the unfolding that happens is immediately irreversible. Now, if you want even more information, Prometheus gives you the chance to correlate this conformational stability, with colloidal stability, and with the aggregation behavior of this antibody at those temperatures. We run this aggregation measurement in parallel, so it's not anything in addition that you have to do, no additional sample, no additional time investment, but you get the information shown here in the bottom panels. On the left, for the first antibody, you can see that for those cycles where unfolding is still reversible, there is no aggregation, but then as soon as unfolding becomes irreversible, there is aggregation. Or more correctly, it's probably the other way around. Because there is aggregation happening, the unfolding is not reversible anymore. The same is true for the antibody on the right. Very early on, we start seeing an aggregation signal that just keeps increasing and increasing with the unfolding of the antibody. So, of course, the fact that that aggregation and unfolding irreversibility are correlated, this is not surprising. This is probably going to be the case for most proteins, but it's still something you might want to know and understand about your protein, especially if you're looking at, for example, an enzyme that has to withstand these kinds of cycles for an industrial application, or if you're looking at storage cycles that happen and you want to know if your protein survives those. Excuse me. All right. The next example, again, is very different. Now we look at chemical stability, and not just simply looking at a delta G measurement. I think you're all familiar with those. They're really useful, really established, but we can go a little bit meta on this and kind of zoom out a little and use delta G measurements at different concentrations of protein to predict aggregation propensity. So this is something that's a bit new, this method. We tried it out in a case study with Boehringer Ingelheim that I will talk about in the next slide, and it worked really well for this one. So if this can really help predict the stability of the protein, that will of course be really useful. The way you do it is, as I said, you measure the delta G for different concentrations of protein, and then you look at the trend that you get. And there's different kinds of trends that you may see shown here. If you get a decrease of delta G with increasing concentrations of protein, this means your protein is aggregating in the unfolded state. If you get an increase of delta G, it means your protein is aggregating in the native state. And if you don't get a trend at all, which is shown here with the green dots, it means there's a lot less aggregation happening because neither your native nor your unfolded state are aggregating. This is, of course, the kind of situation you would want. This is the situation you would look for. And what we did in collaboration with Boehringer was to look at different formulations for a specific antibody and find the best formulation. So we looked at 5 different formulations. You can see them here, F1 to F5, to predict which one would be the best for long-term storage of the antibody. And you can see that for almost all of these formulations, the trend in delta G is negative. So we get a decrease and a downward slope, except for formulation number 4, where all of our delta G values are scattered around 0. So we pretty much have no trend, no significant trend here, and we expect the least aggregation for this formulation. Now, what we did in parallel to these measurements was also thermostability measurements and aggregation measurements, all done on Prometheus, all done in a day, and they all agreed that formulation number 4 should be the best one. And then, of course, as a confirmation, Boehringer used those samples in those formulations to store them for a year or a year and a half, and after that came back to do their normal measurements that they would do with these types of samples, turbidity, monomer content, and such, to check where aggregation had happened, and they also found that formulation number 4 was the best one. So as I said, if you can predict your best formulation in a day instead of having to wait a year or a year and a half to find out which one was the best one, that can save you a lot of time, at least in kind of kicking out those that don't look promising at all. With that, I want to move to the next part where I explain a little bit more about how the technology works. First, let's look at what happens inside of Prometheus. So we use capillaries for holding the sample during the measurement. These are really small glass capillaries, single-use capillaries that hold a volume of 10 microliters of sample, so not a lot at all. And these capillaries are placed directly onto the thermal element inside the system, which does the heating. So this applies the heating ramp or temperature cycles that you want to apply. And then as soon as you start the measurement, these capillaries are scanned every 3 seconds. So the whole tray is scanned every 3 seconds, which gives us a data point every 3 seconds for each of those capillaries, a ton of data points. So this is pretty much what happens. We have a flexibility, of course, in the temperatures we want to apply, in the heating rate that we want to apply, so that we can really design our experiment the way we want. There are, of course, standard settings that people like to use, so this will be scanning from around 20 degrees to 95 degrees and using a heat rate of 1 degree per minute. That's the normal thing that people would do. This kind of measurement would take 75 minutes, and then you're done. Now, what do we even really measure? I've talked before about how we look at the tryptophan fluorescence of the protein, but how do we really do this? So in this image here, you can see the tryptophan fluorescence emission spectra of a protein in the folded state in gray, in the unfolded state in blue, and in a kind of an intermediate or transition state in red. Excuse me. And you can see how this spectrum changes. You can see that the intensity typically decreases upon unfolding, and you can also see that the peak shifts. So while we typically have a peak, an emission maximum peak around 330 nanometers for the folded protein, this moves to around 350 nanometers for the unfolded protein. This is, as I said, due to the changing environment of the tryptophan. And this also means that we can collect all of the information that we need to know about the protein unfolding by looking at these two windows of wavelength. So by looking at 330 nanometers and 350 nanometers, we get everything we need. We call this dual UV detection. Now, the kind of data that this collects, the raw data, so to speak, is shown here in the center panel. Throughout the measurement, we collect the fluorescence intensity at those two wavelengths, which means we get data that looks something like this. Typically, fluorescence decreases with temperature, so that's why we have a general downward slope, and then at some point we get this characteristic transition of the protein unfolding. And what we typically do, though, is we look at not at this data the way it is. We can, but we don't, because we prefer to look at the fluorescence ratio of the signal at 350 nanometers divided by the signal at 330 nanometers, as is shown on the right. This is more robust against things like air bubbles in the sample or fluorescent buffer additives and other things. So we like this a lot. And in addition, as I described before, we can look at the first derivative, which is shown here in blue, if you prefer peaks to transitions. So these are all the different options of looking at your data that you have, and the reason why the data looks the way it does. We use this dual UV detection because it makes us very fast. So this is what enables us to do these fast scans every 3 seconds and give us this high data point density and therefore this high resolution and precision. In addition to unfolding, as I said, we can detect aggregation on Prometheus. This is done in parallel, so it's an optional upgrade you can get for the system, but it happens at the same time on the same samples. What we do is shown on the right here. There is a capillary that does not contain any aggregation, where we shine light in, and the light then gets kind of goes through the capillary. It gets reflected by the surface that the capillary sits on, because that happens to be a mirror surface, and then the back reflected light is detected and used to calculate what happens in the sample, because of course if you have aggregation in the sample, a lot of the light that you shine in will be scattered elsewhere and it won't be reflected back and therefore won't be detected. So that's a really simple concept actually, but it also means we can be really, really fast and get the same data point density and so on, and do this all in parallel. Typically, the kind of data you get out is shown on the left. Of course, you start with a low signal. At some point, aggregation starts happening. This point we call the onset of aggregation, and then the signal increases until the protein is fully unfolded and aggregated. This onset of aggregation is also the parameter that people are typically looking for, and that you would use to compare across samples or conditions. It's typically regarded as a temperature that you want to stay below, of course. And now I want to kind of reiterate a few of those things I've already mentioned that kind of sum up to be the reasons why you might want to choose Prometheus. With Prometheus, you can trust your results thanks to the ultra-high resolution and precision, which in turn is thanks to the unmatched density of data points. So this means that you can really trust that what you see is really happening in the sample, and also that you're not missing anything. You can use Prometheus in any project to address all of your targets because it can work with pretty much any protein, any concentration, any type of buffer. So really for concentrations, we can go as low as 5 micrograms per milliliter, meaning you can use it at those very, very early stages where you have low scales and low yields. But you can also use it with formulation. Concentrations above 250 mg per mil are not a problem. You only need the 10 microliters of sample, so that's not a lot, not a high demand. You can use any buffer, any type of detergent, any type of excipient, whatever you want, which all of these combined mean that you don't have to prep your sample in any way. You can just stick it in and start the run. I've mentioned before that you're flexible in the type of ramp and temperature range you want to apply. The measurement is fast. It needs only the time it needs to heat. That's it. And finally, Prometheus can really help you make your research more efficient because it's an easy-to-handle device. You don't need a lot of training. The software is also really intuitive. There is automatic analysis functionalities, and the instrument is generally maintenance-free. Now, in the images, I just want to say that in the foreground, you see the standard Prometheus system. This is manually operated, but of course, we also offer an automated version shown in the background picture with a robotic arm that handles your samples and loads and so on, so you can really get a lot of hands-off time here. Just put your plates in and walk away. And with that, I just want to very briefly mention that while we do offer the Prometheus to look at protein stability, we also have what we call the Tycho system to look at protein quality, and the Monolith system to look at binding affinity. So if you have any of those needs or interests, feel free to get in touch, and we'd be happy to talk about whether those fit your specific workflow. And with that, I'm done. So I'd like to thank everybody for your attention. Thanks again to Thorsten for the great first presentation, and I'll be happy to take questions.
Thank you, Beate. Now we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. Now, with that, our first question is, you are collecting a large amounts of data on multiple candidates from expression, purification, formulation, and analytics. How do you finally select the best candidate?
I think that's a question going to me, so thank you very much for raising this. It's a question that we discuss also on various conferences because it's a challenge that also other companies are facing in a similar way. So for antibodies, I would say it's typically straightforward because what I mentioned in the beginning is that there we have pretty defined requirements to fit those molecules into our platform processes. So we have well-defined thresholds that we typically do not want to exceed, and if we hit those thresholds, this might trigger then selection or deselection. In the case of all other protein formats, it's a matter of bringing the right people together and really understand the severity that we do see with this. And this also might really strongly depend on the route of administration that the project team is aiming for, also the unmet medical need in terms of what risks and efforts are we willing to go for. As an example, we might have a protein with relatively low yields after the purification process, but we get a high purity and the molecule also shows the respective requirements for stability. There's a question is, what costs are we able to carry to get the molecule still produced when certain small amounts are required only? This might not as a big issue as we would need when we need these molecules also in very high or large amounts and high concentrations. So it really depends on the specific situation of the project, how we then balance and rate the individual factors.
Yeah, I can just comment that of course it would depend on your final goal. So we try to help Prometheus users with the software that we provide, which as I said does a lot of the analysis automatically, so you can get the parameters that you're interested in. And we are very close to actually launching a software that gets you more analysis functionalities, where you can look at different things, plot them different ways to make it easier for you to get the information you need to make your decision.
Thank you for those answers. Our next question is, do you assess multiple candidates on multiple expression systems? What are your decisions criteria when you are screening different expression systems?
Okay, thank you for this question. Yes, this is a very important factor which I showed on the earlier slides, that typically we do have a set of molecules in the screening phase also for therapeutic proteins. This can vary quite significantly. I mean, our extreme cases were just one molecule, but where we then very intensively screen for the respective expression systems to really make sure that we can enable this project. But there are also cases where we then have more than 10 molecules and where we still need to screen for the best expression conditions. So it's a balance between the number of candidates and the expression systems that we are, or vector systems that we are going to screen for. The decision criteria again go back to the specific project needs. So if it's the titer that purely needs to be improved, we of course aim for the highest expressing set of conditions. However, if we also have a combination with potential liabilities with degradation, for example, this also might kick in and needs to be taken into consideration. So there we might compromise on the titer to rather go with a system that does not show the, for example, proteolytic degradation of the molecule.
Thank you for that. Our next question is 22.1 CFR Part 11 software available for the Prometheus system?
Yeah, I think that's a question for me. Yes, it is. Of course, we provide this kind of software for those regulated environments that need to really document their experiments and be able to report it all according to the FDA regulations.
And another one is, can Prometheus analyze proteins without tryptophan residues?
Of course it can. So tryptophan residues are not the only fluorescent residues in proteins. There's also tyrosines which contribute significantly. So if your protein does not have tryptophans but it has tyrosines, then you're fine. If it has neither tryptophans nor tyrosine, then you've run into a limitation of the method. But we hardly ever come across those types of proteins.
And I think with that concludes the question and answer segment. I thank everyone for the questions. Before I finish the webinar, I would like to ask our presenters, Thorsten Lorenz and Beate Kern, if they have any closing remarks.
Beate, would you like to start?
Sure, I'd be happy to. I just want to really thank everybody involved. I think this was a great combination of a real life presentation of what needs to be done in order to develop good therapeutics, but also kind of talk about how we can contribute and help with that. And I'm looking forward to receiving more of the questions and get in touch with people.
Great. Along the same lines Beate was saying, so first of all, I would like to thank everybody again for joining in this webinar and also for putting these questions. I also appreciate it a lot to have this webinar together with Beate, so thank you to you. And my closing remarks are really also, since these were just a few questions that may come up, and also seeing the need generally to really have an extensive exchange on understanding what we are dealing with in the pharmaceutical industry when it comes to new protein formats. I also will be very happy if people would like to reach out and also meeting you also on conference and discuss these in more in detail. So thank you very much and have a good day.
I would like to take this opportunity once more to thank our presenters as well. Thank you, Thorsten Lorenz and Beate Kern, for sharing your knowledge with us. I would like to remind our audience that they can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. Thank you, everyone. Have a great day.